oocyte, what kind of oocyte would this be if it is processing on the way, right? What kind of oocyte would this be? Nope. Secondary oocyte, because remember, right before ovulation, it completes meiosis 1 and enters into meiosis 2. So we have this as the oocyte, there's the nucleus, and this is your corona radiata, and the zona pellucida is this little blue whitish line on the inside. These are important structures for you to remember because they get carried along with the oocyte once it's ovulated and traveling down the oviduct. The sperm have to burn through the corona radiata and the zona pellucida in order to hit the plasma membrane and do something called fast and slow block polyspermy. Because once one sperm hits the zona pellucida and then the plasma membrane, we can't have any more sperm entering into that oocyte. And so things happen to the oocyte that prevent that from happening, which we'll talk about when we get to the lecture. So we go from our secondary oocyte to a secondary oocyte that has had a sperm hit the plasma membrane. This model right here, number 2, used to have a little string hanging off of this little blue dot. This is the head of the sperm, and the tail would have been that little piece of string that was formed. And we have the secondary oocyte nucleus that's going to complete meiosis 2 now. This one has the completion of meiosis 2 and the production of the polar body. So we have a second polar body at the very least, and the nucleus of the sperm. And then we have the 2 pronuclei. So we have a sperm's nucleus and a oocyte's nucleus. They're going to merge after they've completed their disassembling. And when they merge, they form one nucleus, and then we have a structure known as a zygote, a single-celled structure, the first of the embryo process. Our single-celled zygote undergoes cleavage, which is a form of division. This is rapid mitotic division. So we have a 2-cell stage. The 2 divide and form 4 cells, the 4 to 8, the 8 to 16, the 16 to 32, and between 32 and 64, we create something known as a morula. This is depicting the morula. That morula, you'll notice, is the same size cell structure, same size structure as the zygote was. So we pack these cells in. The cells are now not called cells, they're called blastomeres. And ultimately, the cells, those blastomeres in the center of the morula, start producing a fluid. And the fluid pushes the cells out, and the fluid fills a space called the blastocele. And the entire structure becomes known as a blastocyst. And the blastocyst is busy burning itself into the wall of the uterus. And I mean burning, because it's using enzymes to do that. And the blastocyst differentiates into 2 different parts. There's a part of the blastocyst called the trophoblastocyst, and another part of the blastocyst called the syncytio trophoblast. And the trophoblast part is actually known as the cytotrophoblast. I have a slideshow and we'll go through it in a little more detail that way, so you can understand it all. Then, remember I told you that the inside of that blastocyst, called the blastocele, is filled with fluid, but it also develops something called an inner cell mass. And the inner cell mass is you. It's going to differentiate into all the different tissues, ultimately via something called gastrulation, where we create the germ cell layers, ectoderm, mesoderm, and endoderm. And then the rest of this trophoblast, this is all depicting the trophoblast, but this part of it is called the syncytio trophoblast. This is where the enzymes are. It's one cell with multiple nuclei. So it's really producing a lot of enzymes and into, as you see here, the myometrium, where there's good blood supply. this is further stage development. Even further stage developments, now we have some differentiation of our germ cell layers of the inner cell mass. We have something called a yolk sac, and this is the beginning of the amnion. This is the chorion of our trophoblast, and it ultimately will become the placenta. This is, I'm sorry, that's not the amnion, this is the amnion created right here. Again, I'll look at pictures together. This one is depicting the folding of the embryo. So the embryo folds lengthwise and laterally. So because it's a three-dimensional structure. And the folding is where we develop the tail, the head, limbs, the internal structures. And this is all happening within weeks of fertilization. We have further developing placenta structures. These are called chorionic villi. And these little structures right here are bathed in maternal blood. And there's an exchange of nutrients and waste across these capillary beds of the chorionic villi, surrounded by venous sinuses, or you can call them a couple different names. But that maternal blood is pooling around these chorionic villi, and again, you get that exchange of materials, not just with simple diffusion, but there's facilitated diffusion, there's active transport, there's receptor-mediated transport, there's all different mechanisms for moving the important products back and forth. So then we have this one that is showing you really rapidly developing embryo and the developing umbilical cord. This is the yolk sac that is starting to disintegrate. Your amniotic sac, amnion, the membrane. You have your chorion here, and your chorionic villi, and you have the wall of the uterus. One wall of the uterus, one region of the uterus is where this placenta is attached. It's called the decidua basalis, which I think of as the base of operations. And then the other side of this developing embryo is called the decidua capsularis. So the capsule around this developing embryo. And then there's a space, which is luminal, uterine luminal space. And then there's the decidua parietalis, which is the outer wall of the uterus that's not involved with any of this development whatsoever. Ultimately, of course, that space gets eliminated because the fetus fills the entire luminal region of the uterus. So these are all here, and we do have a key for them. There's also another model right here, which is the embryo itself. And I want you to pay attention to it, because one of the things that we don't talk enough about are these structures right here called the somites. The somites ultimately become the vertebrae, and then some ribs and developing structures from the vertebrae. It shows you the pharyngeal arches, and this is sort of linking us to all the other species that live in the water.
Yeah, it looks
where
like a
gills
seahorse.
are, right? Yeah,
like a
and
seahorse.
horses have pharyngeal arches, and so it's a nice way to see a connection between our development and all the other biota. I don't know if you've ever seen those images where, I forget what stage it is, but we look exactly the same as a chicken, we look exactly the same as a rat, we look exactly the same as so many other different species. Because, right, you can't deny our DNA. We were talking earlier about microbe. The bacteria's DNA is constructed in exactly the same way. Not the same series of base pairs, but still the same A to C, A to T, C to G. Can't deny it. We have some, I think we talked about it in the nervous system. We talked about conserved genes, highly conserved genes, like the need to eat, right? Appetite. That is a highly conserved gene, the one that drives us to eat. Because everybody needs to eat. Every species, everything, bacteria eat, everybody eats. When they're really highly conserved. I'm just kind of goofy about that. Anyways, check that all out.